Yeah, okay. We studied RAPD, which is random amplified polymorphic DNA. You can see this is a good picture of the process. I just wanted to share this so that you will be more clear with the concept, as we don't have a picture in the PPT. The blue letters here shows the abbreviation, whereas the black full form shows what is the full form. R stands for random, A stands for amplified, P stands for polymorphic, and D stands for DNA. As the name itself is self-explanatory, we amplify DNA randomly using primers to get fragments. If you can see here, whichever is given in blue color here, like this one, this one, this one, this one, these are primers. These are primers, and you can see the products of primers. If I number them 1 and 2, I have two products of these two primers which are given in a blue color shade, which is different from the others. Now these primers in the direction specific to them, if I take it as 3 and 4, so they will synthesize a piece much smaller than the first two. So I have pieces like this, and for 5 and 6 I have pieces like this. For 7 and 8 I have a longer pieces. Depending upon where the primer is attached in both the strands of the DNA, they start amplifying the template for the distance they can do. Random amplification of DNA by employing primers, universal primers, which act as a molecular marker. Now we perform the electrophoresis for those, and as we can see, the size is from bigger to smaller, and these are the smallest. I see them settling at the last, and the next largest is the blues, and the dark blue, and finally the violet. So for a given fragment of DNA, for the primers used, the amplified segments will give me a definite pattern in electrophoresis, random amplification, polymeric DNA. So definitely, if another DNA is used with the same primers, the pattern will be different. So I get one DNA, one pattern of electrophoresis for the amplified segments. This is how I understand DNA by using this technique. Got it? So here the primers are helping us to amplify fragments which we electrophoresicize to create a pattern to understand that DNA fragment. Primers act as molecular markers here.